So good morning, everyone, and thanks for joining today's seminar. I would like to begin by acknowledging the Wurundjeri people of the Kulin Nation as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining me from today. I pay my respect to their elders, past, present, and emerging, and to the Aboriginal elders of the other communities. My name is Maria Elena, and I'm a CCMMP student. I'm doing my PhD at the UniMilk Node, and I'll be your host for today. I'd like to start with a few housekeeping rules for today, starting by letting everyone know that this is going to be a recorded event that you can find uploaded on the CCMMP website along with our previous seminars. Please make sure that your microphone is muted throughout the presentation and leave your questions to the end of the seminar to minimize the interruptions. And the presentation will be going on for 45 minutes, and then we'll have the 10 to 15 minutes Q&A session. Moving now to our presenter for today, I am delighted to introduce you Dr. Lisanne Spenkelink. Lisanne obtained her joint PhD in biophysics from the University of Grongingen in the Netherlands and the University of Wollongong in Australia. She has recently been awarded an NHMRC Emerging Leadership Fellow at the Molecular Horizons Institute at the University of Wollongong. Her research focuses on the development and use of novel single molecule techniques to study the dynamic behavior of proteins in complex biological systems with a focus on DNA replication, both bacteria and eukaryotes. So bringing together her expertise from the fields of physics, chemistry, and biology, Lisanne aims to use these techniques to evolve sensors for single molecule diagnostics. So today, Lisanne will highlight how single molecule visualization has helped reveal the biological stochasticity using the DNA replication system as an example. So please join me in welcoming Lisanne. Lisanne, the screen is all yours.
Thank you so much for that kind introduction. So when I was invited to give this talk here today, I sort of discussed with Aidan Grosas on what I should be talking about, and we sort of came to the conclusion that it would be nice to maybe share some of the work that we've done revealing dynamics in complex biological systems, and sort of relate that back to what that means for mostly a structural audience. So I will do that. I will use, like you said, DNA replication as sort of an example system, because also that's what we've done most of our work on. And then in the second part of my talk, I will discuss how we use these single molecule techniques to develop new tools to do directed evolution to ultimately evolve new sensors for diagnostics purposes. So if I can get this slide to move, here we are. So just very briefly about myself, I'm from the Netherlands, which is a teeny tiny country right next to Germany and the UK. I did my undergrad in the north of the country here in Groningen. And then just as I had started my PhD in the lab of Antoine van Oijen, he announced that he was moving his lab to Wollongong. So I sort of had a decision to make whether to join him or to stay in the Netherlands. And, you know, the Netherlands is a really beautiful country. So this is a photo of Groningen where, you know, on beautiful nights, everyone likes to get outside and have drinks and, you know, have good chats. It's beautiful. For those of you that have been to Amsterdam, Groningen looks like that too. It has these beautiful canals with the boats and it's all very picturesque. But mostly the Netherlands, the weather looks like that. It is very often very grey and rainy. And the one day in summer where the weather is actually nice, your beaches look like that. So, of course, I then decided that I would love to get away from that. So I took the trip from Groningen to Wollongong, which apparently at the time was $4,000. I guess that sounds about right. And of course, there the weather mostly looks like this. And the many days in summer when the weather is nice, the beaches still only look like that. So I much prefer this over the Netherlands. All right, so for today's talk, I kind of took, I recently saw this clip from House. He says this: how do you see something not move if nothing is moving? And that is sort of really the crux of a lot of the work that we do. We want to reveal all the dynamics that are going on in biological systems. So we know a lot about all of these systems. Obviously, we know a lot about we know a lot through traditional biochemistry assays. We know a lot through structural work. But what we tend to ignore is all the dynamics and the importance of these dynamic behaviors in the ultimate outcome of what the biological system is doing. So how do you see something not move if nothing is moving, which is sort of a backwards way of saying is, how do you see something move if nothing is moving? So we want to visualize movement. And I guess another way of representing that is sort of in explaining why we need single molecule techniques to do this. So on the left-hand side here, we see a square, and we could see that as our traditional biochemical assay where we look at an ensemble average. But what we cannot ever see is, if we don't see dynamics, we don't see if the square is a square, or if it is actually 4 dots that are moving like this. So I should credit my student, Nick, here for making this animation, which I think beautifully illustrates the purpose of doing single molecule assays, where we can actually, in real time, visualize the dynamic behavior of individual proteins within a biological system. So like I already said, I will talk about the single molecule visualization that we do here in Wollongong, mainly focused on DNA replication. I will highlight how we can use these assays to see protein dynamics, and then I will touch upon how that relates to monkeys. And then in the second part of the talk, I will talk about our new stuff, so working on directed evolution and ultimately using that to develop diagnostics tools. So I think a lot of you have probably seen this movie. It was made by Drew Berry more than a decade ago now. It is an animation of the E. coli replication system, or the E. coli replisome. And what you see is on the left-hand side here, your parental double-stranded DNA is being unwound by a helicase shown in blue, which unzips the double-stranded DNA. And then new DNA is being synthesized on the 2 daughter strands. On one strand that happens continuously, and on the other strand, new DNA is synthesized in the form of short patches, which are called Okazaki fragments. And this is obviously a very beautiful movie. And what it shows is that, there is a little bit of movement happening, but where the movie really sort of falls apart is in the way it depicts this in this sort of big, open, empty space. We all know, of course, that that's not very representative of the cellular environment. In fact, this machine, or this system, if you want to call it that, is surrounded by many other molecules, and also by many, many copies of these individual proteins that make up the system, sort of all around it. So we wanted to study what the effect of that was, and essentially answer the question, how does this machine behave when it's not in a big, empty, open space like that? So we have, as in Antoine's lab, have used a single molecule rolling circle assay for more than 2 decades now, I guess. So what it essentially is we use circular DNA templates on which we can then load the helicase and all other proteins, and we can then visualize DNA replication on that. So what that looks like in the form of an animation, you can see here. So the animation of the top here and the actual movie down the bottom, they're both looped. So what you see is that when replication starts, the inner circle forms the template for leading strand synthesis, which you can see in sort of the light blue color. And then this newly formed leading strand can form the template for lagging strand synthesis, which is what you can see in the dark blue color there. We can bind this entire thing to the surface of a microfluidic flow cell, and we can put that on the microscope. And we can then add a fluorescent stain that stains the double-stranded DNA. So when we then apply a flow and continuously provide all the proteins that are required for replication, what you see is this growing tail of double-stranded DNA that is stretched out in the direction of flow. Because this replication keeps going around this circle, in theory, replication should be able to go on and on and on forever until something breaks. Now, obviously down here, I'm showing one molecule, but that's obviously not all we see. This is typically what we see in an entire field of view. So this is what we see in one single experiment. So in the background, you see all these dots. Those are the starting templates that we start off with. So these tiny circles. And then all these lines that you see growing are individual DNA molecules that are undergoing replication and that we can track as a function of time. So even just by looking at this video right here, we can already see again the power of doing single molecule assays. we can see that not all templates are replicating. We can see that some replicate a lot of DNA, some replicate a tiny little bit, some go fast, some go slow, some stall. There's so much information already in this that you would not get from a traditional ensemble biochemistry assay. For example, if you ran these products on a gel, you would see one band and learn not a lot. So if we focus in on one of these molecules, for example, this one, we can represent the movie of this one molecule in the form of a kymograph. So a kymograph is nothing more than putting each and every single frame of the movie for this particular molecule right here, one after the other. So you then have time here on the y-axis and the length of the DNA on the x-axis. So what you see is that replication starts at the top here, and as more and more DNA is being synthesized into this tail, the tail gets stretched out in the direction of flow, and you get this beautiful long product. We can then measure the position of the tip of this DNA, which is where the replication system is. And from that, we can track the position and get information on how much DNA has been synthesized, the rate of synthesis, and the changes in rates along this. So if we do that for all the fields of view, for all the molecules in this field of view, we get this rate distribution. And again, here, it's sort of like really should hit you in the face that there is a lot more information here than what you get from traditional rate analysis of replication. So the average rate that we get of around 450 base pairs per second is very similar to what you would find in other assays. So that's very consistent. But what we can see is that there's actually a distribution, a very large distribution of rates that are happening here. So there are times where replisomes move at almost 2,000 base pairs per second, and there are times where replisomes move at only 100 base pairs per second. And they switch and swap between all these rates. So different molecules move at different speeds. But as you can see from this one example right here, this one DNA molecule gets replicated sometimes very fast and sometimes very slow. So there are these continuous changes that you would not pick up on in traditional biochemical assays. All right, so now that we have this assay, we sort of wanted to use this to not only look at the DNA, but also look at the individual proteins that make up the replisome and see how they behave. So we chose the polymerase. So the polymerase is the actual copying machine of the replisome. So this was work done with Jacob Lewis. So we wanted to see how these polymerases behave. So we put a fluorescent label on them so that we can see them in our turf microscopes. Now, the textbook model is that these polymerases, once they assemble within the replisome, they remain bound, and they remain bound the entire time as they copy the entire genome. So that is consistent with the movie that I showed made by Drew Barry, where you sort of have this one machine almost that starts and copies the entire genome. So that was the textbook model. So, you know, the textbook model can be correct. So when we set up the assay in a way where we assemble the machine and we then wash out all components and then let replication proceeds, so we only feed it nucleotides, no other proteins are around, then you can see that replication happens as per normal. So what you see here is a movie showing the signal from the polymerases only, so we can't see the DNA here. And replication starts at the bottom, and as the polymerases are synthesizing DNA, you get this tail that grows up in the direction of flow, which is now from bottom to top. And here you can see the corresponding kymograph of this movie. So that's all the frames of this movie put side by side. And what you see is that replication starts here, and the polymerases move with the tip of the DNA until eventually you see that signal disappear. And that is because the fluorophores that are on these polymerases, they photobleach. Photobleaching is irreversible. It's sort of a chemical change in the fluorescent molecule. It just damages, and it will never ever come back. But replication still continues. So the polymerase itself is still fine. It's just its fluorescent label is dead, and we can't see it anymore. But replication continues well after this movie ends. So, you know, the textbook can be correct. But you can probably guess from the way that I say can be correct that we also found many situations where the textbook wasn't correct at all. So when we have other polymerases in solution, so when we mimic more the cellular environment where we have high concentrations of replication proteins around, so we don't wash them out, then this is what we see, which obviously looks very, very different to what I showed you on the previous slide. So again, here we have the movie on the left with the DNA moving up in the direction of flow. And it's probably better visible in this chymograph here. There are two very striking observations here. First of all, we see these lines that are sort of in the wake of the replosome. These are polymerases that are being left behind. So they they formed part of the replosome at some point, but then they were left behind at unfinished Okazaki fragments. And we have shown that is indeed where they are. So they sort of finish an Okazaki fragment on the lagging strand and then they just sit there. Probably more importantly, though, is if you focus on the polymerase signal at the tip of the DNA, you see that it doesn't photobleach. And trust me, we have tried, but it does not photobleach. So what we sort of inferred from that is that, you know, the polymerases that are here at the tip of the DNA, they must exchange before their fluorophore can photobleach. So exchange must happen faster than photobleaching. To sort of quantify that, we measured the intensity of the signal at the tip of the DNA, which is what you can see right here. And then we on the same graph plotted the photobleaching lifetime, which is this one right here. So you see that, you know, if the same polymerase were present here the whole time, it should have photobleached about halfway through the movie. There's absolutely no chance that it could have lived that long. So that must mean that we get very fast exchange of polymerases. They exchange so fast that they don't have time to photobleach. So then we wanted to know, well, how fast is fast? And we thought we'd be clever. And we use two colors to see this exchange. We thought if we have half of our polymerases labeled with this magenta color that we had before, and we label the other half in a green color, then if it exchanges, we should see magenta swapping for green, swapping back for magenta, and so on. Well, that's not entirely what happened. So we did this experiment, and this is what it looked like. So here you see the movie where we have an overlay of the two colors. So we have the one population labeled in magenta and the other labeled in green. And again, replication worked just fine. And here's the chymograph of that. But, you know, this expected result where we saw one color swapping for the other, that is really not happening. We actually see mostly both colors there at the same time always. So that just means that the polymerases were exchanging so fast that we could not resolve their exchange. So the exchange dynamics were so rapid that we had no way of resolving it. So we then changed the conditions again. So here we have a concentration of labeled polymerases that is similar to the concentration that you would have in the cell. We thought, well, we now have the two extremes. We have this condition where exchange is very rapid. We have the other extreme where we have no polymerases in solution, so exchange can't happen at all. What if we go somewhere in the middle and we go at a low concentration of polymerase in solution? And it was then that we saw our expected results. So if you look at this chymograph, you see that we have magenta exchanging for green. Sometimes both are still present at the same time, but then we go back to magenta, to green, to magenta, and so on. So what we concluded from this is that these dynamics that we see, this rapid exchange, is dependent on the concentration in solution. And when you think about that initially, that's a bit counterintuitive, right? How can the on rate or off rate, how can the off rate depend on the concentration in solution? That is a bit weird. So this is where the monkeys come in, and we tried to explain this by sort of using this analogy of the monkeys being the proteins. And the monkeys have at least two binding sites. One binding site is on the branch, and the other binding site can be on the bananas. So if we start on the right here, where there is only one monkey, so that is equivalent to the situation where there are no polymerases in solution, so there is no competition for any of the binding sites. If the monkey lets go of the bananas, that's fine. It's still maintained within the system, and it just hangs on to the branch, and it can just grab the bananas again, no issues. However, if there is another monkey close by, aka if there are other polymerases in solution, then if the first monkey lets go of the bananas, so if it lets go of this weak binding site right here, then there is another monkey so close by that it can just grab the bananas. And then at that point, this poor first monkey is only holding on to the system with this one weak binding interaction on the branch. And of course, eventually that will dissociate. And that is how you can have very stable binding in one scenario versus very dynamic exchange in the other. I've showed you today the example for DNA polymerases, but we have shown the same for single-strand DNA binding proteins, for beta sliding clamps. Almost everything in the repliposome exchanges in this way. So I guess this initial movie that Drew Berry made, no matter how beautiful it is, it does require some updating. It is a lot more messy than we initially thought, and our single molecule assays have taught us that. So why does this matter? Why do we care about if it exchanges or not? Why do we care about if it's stable or not? So to answer that, we sort of looked into some downstream effects, and we started to probe the system to see if we get different behaviors depending on the exchange dynamics. So this is work done by a former PhD student in the lab, Gerline Kaur. So she challenged the replication system by adding in RAR-A. What RAR-A exactly is doesn't really matter for this presentation, but essentially what it does is it disrupts lagging strand synthesis temporarily. So when you do that with polymerases in solution, so when you have the option for exchange, this is what you see. And if you think that that is underwhelming, then I agree. You can't really tell from this chymograph that we even added RAR-A. You'll just have to trust me that it's there. But from this chymograph, you see that replication just starts as per normal. You get this long tail of growing DNA, and everything looks happy and fine. However, if we now go back to the situation where we can't have polymerase exchange, so we have no free polymerases in solution, then suddenly the entire situation looks a lot different. What we see is we get these huge single-strand DNA gaps, which you can see in this chymograph here by this black area. So the fluorescent stain that we use, it only stains double-stranded DNA, not single-stranded DNA. So we get these gaps that are very, very large. In this particular chymograph, it's about 20 kilonucleotides, so 20,000 nucleotides. But we've seen examples, for example, here where it's almost 80,000 nucleotides, and these products are full of them. So I guess what we conclude from that is that polymerase exchange gives the repliposome adaptability. It allows it to adapt to things like stress or challenges or things like that. Whereas if you only have one set of polymerases at the start and you don't have a way to adapt, then all sorts of bad things start to happen. And of course, in our assays, this is fine because we're in an in vitro environment. We have fully reconstituted our systems. There is nothing that can attack this single-stranded DNA. But having single-stranded DNA in a cell is like really bad. Everything wants to attack single-stranded DNA. So this is not a situation that you would want to encounter in a cell. So this is one of those examples where polymerase exchange really helps the repliposome be more robust and adaptable to whatever challenges you might throw at it. All right, so where do we want to take that from here? So we want to sort of go with that, and we want to explore how these dynamics of all these repliposome components affect other processes happening on DNA. So a PhD student in the lab, Michelle Sharma, is looking into replication transcription conflicts. So he has set up an assay that, as far as I know, for the first time allows us to study replication transcription conflicts in real time using fully reconstituted systems. So where we have the reconstituted repliposome and then also purified RNA polymerases, and we see the two active complexes hitting each other and inhibiting each other. And we're sort of really interested in seeing how these conflicts get resolved and how polymerase exchange dynamics play a role in that. We also are looking into supercoiling. So this is work by Elise Wilkinson. All our work thus far, and almost everyone's work looking at in vitro reconstituted systems, even in bulk assays, ensemble classical biochemical assays, is done on relaxed DNA templates where there's no constraints. Which is, again, not very physiologically relevant. Inside a cell, DNA looks more like this. We know that it forms all these secondary structures and loops around itself, almost like a phone code right here. So Elise has set up an assay that allows us to do that, which is what you can see in this movie, where these loops are sort of moving around the DNA. And we're interested in seeing how this affects the DNA replication, essentially. And then finally, we're looking into G quadruplexes. So these are secondary structures that can form in DNA. We know that they can block DNA polymerases. This is very well established. But what we don't know is what happens to these polymerases. And now that we can think about the fact that polymerases are exchanging all the time, you know, what happens when they hit a roadblock like that? Do they still exchange? Do they remain bound? And then, obviously, what does that mean for when eventually this G quadruplex gets removed? Do the polymerase dynamics change the outcome of the situation after that? So Nick is really interested in looking into that. So lots of stuff going on there. I guess now that I've talked about half an hour to you guys about how exchange is very important and dynamics are very important, and how we should be thinking about dynamic behaviors all the time, and you know, how can we see if something's moving, if it's not moving, what does that then mean for structural biology? So I obviously don't want to say that structural biology is not important. It is equally important in my view. So you know, I think there's a lot to learn by combining these two, by combining single molecule techniques and looking at dynamics with structural biology, and sort of figuring out the different structures along the way during the dynamic pathways, during the dynamic changes, and see how the underlying structure determines these dynamics, and the other way around, how the dynamics change the structure of the proteins. And we need both of these things. So while I am sitting here talking about the importance of dynamics, it is equally important to know the structure, the underlying structure of the things that are happening. All right, then for the remainder of my talk, I want to move on to part 2. So this doesn't have a whole lot to do with DNA replication at all. So this is a new program that we started about 3 years ago in the lab, where we're looking at doing directed evolution at the single molecule level. So directed evolution is a technique that's existed for decades, and it's helped us as a society very well. You probably are aware that it's the process of where you grab a natural gene of a natural protein that exists, and you go through rounds of evolution in the lab until you have improved your protein and you get a desired gene of interest out that does something for you. A very relevant example is PCR polymerases. Polymerases obviously exist in nature, but we have improved them, and they are now what is the gold standard for, say, COVID tests, right? So we have improved them and made them better, and they now work for us in a biotech application. So these techniques exist, but they do not allow you to select and evolve based on what I call single molecule traits. So these are the traits that I've discussed previously. So things like dynamic behaviors, short-lived intermediates, on and off rates, stuff that you can't see with classic traditional biochemical assays, but we now know are very important for the function of these molecules. Single molecule measurements can, of course, measure these traits. Like, none of this is very hard to do. There are many labs in the world that can measure these traits at the single molecule level. But what was challenging so far is, you know, say that you have found in this field of view right here in the middle that this particular spot does what you want. How do you then know what variant in your very large library of variants, how do you know which one that is? So essentially, if we represent it as this group of people, how do we know who the orange guy is? And that is something that we tried to address. So we have developed a single molecule genotyping assay, where we directly link the genotype to the phenotype to then identify a single variant in amongst a large library of variants. So essentially, we're now able to answer the question, who is this orange guy? So it started off with this idea that, you know, when you create a library of genes, so with all different variants, and you then express their proteins, you sort of traditionally lose that connection between DNA and protein. So even if you then find the protein variant, it's very hard to link that back to the original genetic information. So we have developed a way to directly link genotype to phenotype, so that link is always maintained. And this is how we do that. So we design these gene constructs that have your variant gene. So within a library, this variant would always be different. Then we have the gene for a snap tag. And we also have these two indexes, which I will come back to later. And then we have a benzoguanine moiety at the end. So we can express this gene in a in vitro expression system. And that takes about a day all up. And what happens is you express your protein variant and you express the snap tag. And the snap tag will immediately bind its ligand, which is the benzoguanine, and create a covalent link between your gene that the protein came from and the protein variant itself. So that is how we create this direct link between genotype and phenotype. So then we obviously need to be able to read out this genotype. That's the entire challenge, right? And this is where the barcodes come in. So we attach to each of these genes, as I showed here, these two indexes, which make up a DNA barcode. And each index can bind a fluorescent probe in solution. Now what we do is we make these indexes very, very short. They're only 7, 8, or 9 nucleotides long. And at those lengths, the binding or the annealing, if you will, of the fluorescently labeled oligos in solution to the indexes is transient. So you get these very short-lived annealing events. And also at these lengths, so 7, 8, and 9 nucleotides, the length of the oligo really determines the duration of this binding. So a 7 oligo will only bind for like a second or something like that, whereas a 9 nucleotide oligo will bind for 30 or even 60 seconds. So there's this huge dynamic range that we can actually use to then identify which length we're looking at. So if we focus in on this one single molecule right here, you can see that this is what it looks like. You get this on and off binding. So you see a high signal when a probe is bound and a low signal when there's no probe bound. And we can measure the duration of this, the time where it remains bound, and that's our on time. And like I said, that very heavily depends on the length of the index. We can make distributions of that and then identify it as a 9-mer in this particular case. So that was only just one example, a 9-mer, but we obviously have in this example 2 indexes present in this barcode. And what we can then do is we can set up this sort of, I guess, alphabet almost of 9 different barcodes that we can read out. So for example, the population down here shown in purple has a 7-mer for index 1 and a 7-mer for index 2. The population of the top right here in green has so you can see the dwell time for index 1 on the x-axis and the dwell time for index 2 on the y-axis. You can see that it is a 9-mer and a 9-mer. So this way we can very easily build up these statistics about where these clusters should lie. So we can train a model with this and successfully have it recognize this training data set. But then, does it really work is the question, I guess. So here we did an experiment where we deliberately left out the variants that sort of lie on the diagonal here. So the 7-7, the 8-8, and the 9-9. And then we asked ourselves, well, can we identify the correct variants? And what you can see really is that there are like a few mishits here and there, but we have a larger than 90% accuracy of identification. And we can visualize hundreds of molecules per variant. So for each of these clusters, we get hundreds of molecules. We can, this reaction, the good thing about it is that it'll just keep going. There, until you hit stop on the recording, it just doesn't stop. So the probes will just keep binding and unbinding the whole time. So we can make this more accurate by just measuring longer. And you can see that we get really good numbers for each of the variants that are present. All right, so I've been showing examples with only 2 indexes because that is sort of visually a lot easier than increasing it. But how scalable is this? How many variants can we actually identify? And to sort of explain that, we make the analogy with letters, words, and letters, numbers, and words. So we have identified 6 unique sequences, which we call A, B, C, D, E, and F. And each of these we have in 3 different lengths, so 7, 8, and 9. We've designed them in such a way that they really can't talk to each other. So a B sequence would never bind to an A sequence, and an A sequence would never bind to an F, and so on. They're completely orthogonal. So if we say that these sequences are letters, and then if we say that each index is a word, so you can have an A7, that's one word, or a C8, that's another word. So you can mix and match the words here in these 2 indexes. Then using that, we can already distinguish 270 variants. So that's all the words that you can see here, all the sentences, I guess, that you can see here. So these are 2-word sentences, right? If we add another index, okay, if we add just one more word, then we can already identify more than 2,000 different variants. So it very rapidly increases. If we add a 4th index, then we can already uniquely identify more than 40,000 different variants. And here you really start to get into the territory of being able to do actual directed evolution, where normal directed evolution studies sort of start with a library size of around 10,000 to 100,000 different variants. So here we're really in the territory of being able to do that, which is really amazing. So we are now at a point where we would love to start doing actual directed evolution. We've got a few trials going. What we want to do in the future is marry this work, the evolution, with some other work that we've been doing at the same time, where we've developed a single molecule sensor that can detect proteins, essentially. So this is work that's been done mostly by Jordan and Connell in the lab in collaboration with Professor Haibo Yu here at UW and Nihad. They are the computational team. So the way this diagnostics tool works is it is based of a nanobody. A nanobody is a small antibody fragment, so it can recognize a target. And then we have added a fluorophore to it. And this fluorophore is quenched when the antigen is not bound. So it is quenched by interacting with tryptophans that exist on the nanobody. And that sort of reduces its fluorescence. And that's why it's called a quench body. When the antigen then binds, this fluorophore is displaced. It is sort of more free in solution. It moves away from those quenching tryptophans and it becomes fluorescent. So you can detect antigen binding by an increase in fluorescence. So this is how we read that out. So this is not at the single molecule level at this stage, but we can express these quench bodies in a similar way in an in vitro expression system, label them, and then when we add the antigen, we get an increase in fluorescence, and we can sort of measure that increase by comparing it to our control, which doesn't have the antigen, giving us what we call a fold sense, which is a measure of how well it works, essentially. So I'm skipping over a lot of work right now, but essentially quench bodies already existed for small molecules, but they didn't really work very well for protein detection. So the team has, through computational work and a lot of experimental work, optimized these quench bodies, placed the tryptophans in optimal positions to increase their performance. And here I show the example of MBP, so maltose binding protein, which is obviously not a very interesting clinical target, but it is sort of a nice protein to start working with and trying to detect. And they were able to get a performance of these quench bodies of 1.46. So you can see a titration here where we measure the the fold sense, so this performance, going from no antigen all the way up to 500 nanomolars of NBP, and you see that you get this beautiful increase in fluorescence depending on the concentration of your antigen, with significant detection down to low single-digit nanomolars. And what's great about it too is that the KD that we measure by this fluorescence detection is 14 nanomolar, which is similar to the KD, very, very similar to the KD of the nanobody. So before we turned it into a quench body. So we haven't really altered the binding capacity of this nanobody by turning it into a proper quench body. So that was great. So what we then did is we used this optimized scaffold, this optimized quench body, which can now be used for detection of proteins. And we asked it, well, can we now evolve this, like start off from this scaffold, but then evolve it to detect something a little bit more useful? And what we chose is interleukin 6. Interleukin 6 is an inflammatory marker that in a healthy human is present at very, very, very low amounts in human blood serum, almost to a point where you can't detect it. And then when inflammation happens, you get increased levels of interleukin 6. So it's sort of an interesting target to detect inflammation and diseases related to that. Our collaborators in Paris did that. They went through 2 or 3 rounds of evolution, starting off from our scaffolds. And you can see the results here. So these are now our performance, so our fold increase for this newly evolved quench body. And you can see that it not only works, but it has also improved the performance, the evolution. And we get detection, significant detection of interleukin 6, again down to single-digit nanomolar. So this is really, really promising. We're not quite there yet that we can detect interleukin 6 in healthy human blood serum, but we can definitely detect interleukin 6 when it is upregulated, and we can detect it in these complex biological samples as well. So that's really promising. So this is what we want to next do with the single molecule directed evolution. Now that we've seen this promise by just doing a traditional directed evolution study using our scaffolds, we can only imagine what we can learn if we do a single molecule directed evolution study. We hope that we can not only get this significant detection limit sort of down to the level where we can detect interleukin 6 in healthy humans, but also see if we can increase the performance even further, because obviously that just increases your level of certainty by quite a lot. So that's where this is going next. We're going to marry the 2 projects, so doing single molecule directed evolution on this. And also we are very interested in sort of using these quench bodies on any target. So if anyone has a target in mind that would benefit from having a quench body that can detect it, we'd be very, very interested in talking to you. So I guess with that, I want to thank everyone that's been involved with all of the work that I talked about today. It's a bit too long to go through everyone, so I just want to mention Stefan, Dave, Jordan, and Connell, and Gerline, and of course Antoine, and also in particular Jacob Lewis, who is now back here at UOW, and we have funded PhD positions available. So if you are anyone or know anyone that might be interested in doing a PhD in sort of Cryo-EM and single molecule detection of DNA replication, then please come speak to us. And thank you for your attention today.
Thank you, Lisanne. Very insightful talk. So I guess that we can now open the seminar for questions from the audience. And if you have any questions for our speaker, you can raise your hand, and you can also type questions in the chat box. I can read them. And I think Inakshi is raising her hand. So if you'd like to unmute yourself.
Hi, Lisanne. Beautiful talk. Can you hear me?
Yes, I can.
Yeah. I was just like, this might be a vague question. During the exchange of polymerase, is it possible that there will be some discontinuities on both the leading and lagging strand? Like, I'm just thinking, if the exchange is happening, then the replication process might be interrupted during the exchange. And is it possible that the leading strand might also have some discontinuous phases like the lagging strand, which will oppose the textbook semi-discontinuous DNA replication, like the hypothesis. Would you like to say something on that?
Yeah, so we've thought about this a lot. So the way that would show up in our assays, so if leading strand synthesis is disrupted, that would look like a pause. So the product would no longer be growing. We obviously see pauses quite a lot. These are things that happen all the time, but it doesn't really seem to depend on the rate of exchange. So these pauses happen when we have absolutely no exchange. These pauses happen when we have very rapid exchange, and the pause duration or the frequency of pauses, nothing seems to depend on the exchange rate. Similar with the rate of synthesis, you can imagine that if you have a lot of exchange, then maybe you expect that that the rate of synthesis slows down because maybe you can't synthesize while you're exchanging. Again, we don't see that. So we've never been able to see an effect of exchange on any of these characteristics. The only way where we see a change in pause duration and pause time is in some instances when we challenge the replosome. So the RARA was a perfect case where we didn't see that, but we can induce certain roadblocks on the DNA where we see stalling. So that's the only time we see any significant change.
Can I ask one more?
I'm fine with it. Yeah.
So you have said that you have observed some pauses in the leading strand as well. Do you think this might be physiologically something significant, or like, I'm just worried that if the second polymerase, like after the micro-milliseconds after the exchange happens, if it cannot bind to that exact location or something like that. Would you like to say something on that?
So yes, I think it's physiologically relevant, but it is hard to say how. I think it just highlights that this stochasticity is happening all the time. Sometimes replication goes fast, sometimes it goes slow, and that is something that is happening all the time and that we should be thinking about. Exactly how it matters in a physiological setting is very unclear at this point, and this is why we want to now look into what happens when you challenge the replosome, and then see if there is any physiological relevance to that. I think the answer is yes, but how, I cannot tell you.
Thank you so much.
We've got a couple of questions in the chat box. So from Uxauti, I'm sorry for the mispronouncing probably. Is there any limitation with the molecular weight of the protein?
Any limitation on what?
I guess probably on the quench bodies. Would you like to follow up with this question? It's UQZZAOTI. Would you like to follow up with another question, and maybe we can move to the question from Thomas? So Thomas asks, do you think the quench bodies could be used for quantification of, for example, IL-6, or is it only usable for detection?
No, I think that ultimately it could be used for the quantification of it. As you saw in my in the curves that I showed, the intensity of the fluorescence in an ensemble measurement, the total intensity depends on the amount of IL-6 that is present. Ultimately, if we can transition this entire thing onto the single molecule level, then the number of binding events that you would see would depend on the amount of IL-6 that is present. So yeah, no, it's not a yes or no kind of answer. It's really a quantification of the amount of IL-6 present.
Great. Do we have any more questions for Lisanne? Reminder that you can raise your hand or write in the chat box. In case no one is asking, I
can guess the limitation of molecular weight. I'm assuming that is on the directed evolution and this sort of linking genotype to phenotype, expressing it in an in vitro expression system. I'm thinking no. We are currently limited because we buy our construct as G blocks from IDT, and they have a limit. So that's where we're limited to at the moment. But no, in principle, these expression systems work with plasmids of any size. There might be differences in expression level, I mean, there will be, obviously, so that might be a challenge, but I don't think that is necessarily linked to molecular weight of the protein.
Ooh, thank you.
Hi, Lisanne. I have a question. I'm sorry, I can't figure out how to raise my hand. It's James Bauer.
Hi, James.
Sorry. In regards to the polymerase exchange, you said you were unable to resolve that time frame, that it happened so quickly.
Yeah.
Just kind of wondering, what's the frame rate on your scans, and what do you think you can improve that to where you could actually see single molecules jumping in and out?
The data that I showed you was done at 200 millisecond frame rates. We have, I didn't talk about this today, but we have actually then quantified the actual exchange time. So we have used a technique called FRAP, fluorescence recovery after photobleaching, but at the single molecule level. So where we very rapidly bleach all the signal in a field of view and then watch the recovery as new polymerases exchange in, which is a far more accurate way of actually characterizing the exchange time. And we've seen that it is, yeah, it's super fast. It's less than 200 milliseconds.
Thank you.
At these high concentrations, obviously.
Right. Thanks.
We also have a follow-up question from Thomas after the quantification via detection question. So he asks, do you think that QuantBody could therefore be in the future an alternative to ELISA, or will it be more expensive than ELISA?
You are asking exactly what we are doing. Yes, we think it could be an alternative to an ELISA, and it will be far cheaper than an ELISA as well. Yes, it really is. It's not quite as easy as just adding your QuantBody. It's a little bit more complicated, but it is a lot faster and a lot faster to do the detection, and a lot cheaper to make the QuantBodies, because we can do this in vitro expression. We're not waiting for antibodies to be produced. So that makes not only the evolution to make your specific recognizer, it makes that a lot faster, and then the ultimate production a lot faster too. So when you add that all up, yeah, it becomes a lot cheaper than an ELISA. So that is something that we are definitely thinking about.
Great. Do we have any more questions for Lisanne? Because I do have a question, and I'll probably take it back to the initial slides that you were showing us, the different DNA templates and the replication. So I was wondering, did you guys check all these different templates that were replicating in different rates, and did you notice the same exact exchange procedure happening? Yeah,
the exchange is really sort of pretty similar across all these templates. Of course, again, there's stochasticity there too, right? So sometimes a polymerase will hang around a bit longer, sometimes it will be much shorter. So even within the exchange rate, it's not a single rate, it is a distribution of rates. But that distribution didn't seem to change depending on what particular molecule in the field of view you're looking at, or how fast the molecule is moving. We've looked at, well, you know, if a molecule is moving fast, does that mean replication exchange rate is slow? No, there was no such correlation.
Yeah.
So yeah, we looked into it, but no.
So no correlation with the sequence either, not length-wise, but like specific nucleotide sequences.
So our assay doesn't have like single nucleotide resolution, but we think that if there was a sequence specificity, then we would see things like pauses or things like fast rates or exchange events always happening at the same lengths of our product. And again, we don't see that. So no, we don't think it's sequence related, but we don't really have the resolution to say yet.
Yeah. Any more questions? We still have a few more minutes if like someone was jumping and ask a question. If not, I guess that we can like conclude for today. So on behalf of everyone here, let's thank Lisanne for joining us and sharing her inspiring research. Thanks for everyone who joined the seminar, and we really hope to see you all again in the next one.